Welcome to BioGuru, and today we are going to discuss about one of the most important biochemical reactions, which is cellular respiration. Meet my dog, Didi. Just like all living things, he needs energy to run around the house and break stuff. But where and how does he get all that energy? This process is through cellular respiration. Cellular respiration is the process of deriving energy from what we eat or from our fuel. It is a set of or series of metabolic reactions and processes that take place in the cells of organisms to convert biochemical energy from nutrients into energy in the form of adenosine triphosphate and then release other waste products. There are stages in cellular respiration, namely glycolysis, Krebs cycle, and electron transport chain.
Glycolysis is a metabolic process that serves as the foundation for both aerobic and anaerobic cellular respiration. In glycolysis, glucose is converted into pyruvate. The first step in glycolysis is the conversion of D-glucose into glucose 6-phosphate. The enzyme that catalyzes this reaction is hexokinase. The second reaction of glycolysis is the rearrangement of glucose 6-phosphate into fructose 6-phosphate by glucose phosphate isomerase. Phosphofructokinase with magnesium as a cofactor changes fructose 6-phosphate into fructose 1,6-biphosphate. The enzyme aldolase splits fructose 1,6-biphosphate into two sugars that are isomers with each other. The enzyme thiophosphate isomerase rapidly converts the molecule dihydrocytone phosphate DAH. Enzyme thiophosphate isomerase rapidly interconverts the molecules dihydroacetone phosphate and glyceraldehyde 3-phosphate. Glyceraldehyde 3-phosphate dehydrogenates and adds inorganic phosphate to glyceraldehyde 3-phosphate, producing 1,3-biphosphogycerate. Phosphoglycerate kinase transfers phosphate group from 1,3-biphosphogycerate to ADP to form ATP and 3-phosphoglycerate. The enzyme phosphoglycerate relocates the P from 3-phosphoglycerate from the third carbon to the second carbon to form 2-phosphoglycerate. The enzyme enolase removes molecules of water from 2-phosphoglycerate to form phosphoenolpyruvic acid or PEP. The enzyme pyruvate kinase transfers a P4 form from phosphoenolpyruvate or PEP to ADP to form pyruvic acid, and ADP result in 10th step.
Krebs cycle. Krebs cycle, also known as citric acid cycle or tricarboxylic acid cycle, is a stepwise cyclic process which is used to oxidize the pyruvate formed during the glycolytic breakdown of glucose into carbon dioxide and water. It also oxidizes acetyl-CoA, which arises from breakdown of carbohydrate, lipid, and protein. The actual Krebs cycle begins when acetyl-CoA enters into a reaction to form citric acid. This cycle was discovered by British biochemist Sir Hans Krebs. The first product of Krebs cycle is citric acid or citrate. Therefore, it is also known as citric acid cycle. Sometimes, Krebs cycle is also referred as tricarboxylic acid cycle or TCA, as the process is tricarboxylic in nature. In eukaryotes, Krebs cycle operates in matrix of mitochondria. It is absent in prokaryotes. Its substrate acetyl-CoA is entrant or connecting link between glycolysis and Krebs cycle. The oxaloacetate acts as acceptor molecule.
Electron transport chain is the last component of aerobic respiration and is the only part of glucose metabolism that uses atmospheric oxygen. Electron transport is a series of redox reactions that resemble a relay race in that electrons are passed rapidly from one component to the next, to the end point of the chain where the electrons reduce molecular oxygen, producing water. There are 4 complexes composed of proteins, and the aggregation of the 4 complexes together with associated mobile accessory electron carriers is called the electron transport chain.
Complex 1 establishes the hydrogen ion gradient by pumping 4 hydrogen ions across the membrane from the matrix into the intermembrane space. Complex 2 receives FADH2, which bypasses complex 1 and delivers electrons directly to the electron transport chain. Ubiquinon, or Q, accepts the electrons from both complex 1 and complex 2 and delivers them to complex 3. Complex 3 then pumps protons through the membrane and passes its electrons to cytochrome C for transport to the 4th complex of proteins and enzymes. Complex 4 finally reduces oxygen, and the reduced oxygen then picks up 2 hydrogen ions in the surrounding medium to make water.
Cellular respiration can occur both aerobically or anaerobically. During aerobic cellular respiration, glucose reacts with oxygen, forming ATP that can be used by the cell. Carbon dioxide and water are created as byproducts. Glycolysis is the process in which one glucose molecule is broken down to form 2 molecules of pyruvate. Krebs cycle takes place in mitochondria, consuming oxygen, producing carbon dioxide and water as waste products, and converting ADP to energy-rich ATP. Electron transport chain is a cluster of proteins that transfer electrons through a membrane within mitochondria to form a gradient of protons that makes ATP.